So keeping the schedule, trying to. Our next speaker is Nieng Yan from Princeton University, or Nieng Yan, sorry, who is now going to talk about membrane channels, ion transport, structural basis for the initiation of electrical signaling.
Thank you, Guy. For the introduction, I would like to thank the organizers for putting together this wonderful program and for inviting me to be part of this celebration. I feel very honored. And after this morning session, I changed my title to a more provocative one, from the structural basis for the initiation of electric signaling to the a mission impossible by today's AI, that is to reveal the molecular choreography of voltage-gated calcium and sodium channels. I have to because like Ron McKinnon will be a speaker, will be the last speaker of this session, I have to admit that, inspired by his pioneering work in the structural biology of voltage-gated potassium channels, and also intrigued by the electric signaling in multicellular animals. Since day one of my independent research program, we have been focusing on the structural analysis of voltage-gated sodium and calcium channels, which are responsible for the initiation and propagation of action potentials, especially the sodium channels. So we all know that electric signaling is critical for the rapid response to signals over a long distance. So sodium and calcium channels, together with potassium channels and the Nobel-winning TRIP channels, all belong to a superfamily known as the voltage-gated ion channel family. Compared to the homotetrameric or dimeric organization of potassium TRIP channels, CAV and NAV channels have this unique architecture of a single chain organized into 4 homologous repeats. Nevertheless, as a structural biologist, I have been focusing on 2 fundamental questions regarding these channels. First is the molecular basis of ion selectivity, why sodium channels selectively permeate sodium ions but not potassium or calcium ions, vice versa. And on the other hand, a more intriguing question regarding this superfamily is probably the molecular basis or the structural basis for the voltage-dependent gating, voltage-dependent activation and inactivation of these channels. So as just mentioned, calcium channels and sodium channels are very unique in that they are made of a single chain. So the transmembrane region contains 24 helices, just as the other members. And these segments are organized into 4 repeats, each containing 6 transmembrane segments, known as S1 to S6. So the 4 sets of S5 and S6 together enclose the central pore domain that is responsible for the selective ion, for the gated and selective ion conduction. And the first segments in each repeat constitute the so-called voltage sensing domain. Pioneering work from Ron McKinnon and others have, revealed a general picture of how these channels work. Basically, the central pore domain contains a selectivity filter on the extracellular side that discriminates different ions, and the intracellular gate responds to the either voltage or in the case of TRIP channels, respond to ligands and temperature and the mechanical force for opening or closing of the gate. And voltage sensors contain this unique gating charge residues that move based on the fluctuation of membrane potential, and the change of the voltage sensor is coupled to the gating of the pore domain. So this process is known as the electromechanical coupling. So our overarching game is to capture the representative calcium and sodium channels in different working states. So I always said that I would like to be a movie producer to make a movie of these channels in action to reveal their complete working cycle. So compared to the homotetrameric VGIC members, disorders. There are several questions that are unique for eukaryotic sodium calcium channels because of their single chain arrangement. So the most obvious question is, how are these 4 repeats organized? If they all observe a so-called domain swapped architecture, theoretically there are 6 ways to arrange the pore domain and voltage sensors. So there has been studies using toxins, but before the structure was revealed, this remained to be an open question. And in terms of sodium channel, its selective filter is quite unique in that 4 different amino acids, DEKA, together define sodium selectivity. This is a different case in the homotetrameric channels, of course, and even in the case of eukaryotic calcium channels, it is 4 carboxylate from glutamate or aspartate that together define calcium selectivity. So how these 4 distinct residues select sodium remain to be investigated. And also as a result of the asymmetric the sequence asymmetry, their activation and inactivation are not synchronized. So it is known that it has been shown that in case of sodium channels, the first 3 repeats activate much faster than the 4th repeat. In fact, activation of the 4th repeat in sodium channels lead to another important mechanism, that is the fast inactivation. So action potentials occur in waves. So to ensure repetitive firing of action potentials, sodium channels have to inactivate rapidly upon its activation. This process can occur just within 1 millisecond. Pioneering work by Clay Armstrong, Francesco Bazzania, and Bill Cattrall have identified the intracellular segment, particularly the segment that connects repeat 3 and repeat 4, was responsible for fast inactivation of sodium channels. In particular, a motif consisting of 3 hydrophobic amino acids, IFM, is known as the fast inactivation motif and responsible for fast inactivation. A ball and chain model was proposed to account for this rapid inactivation process. However, this model contradicts with some functional analysis. Therefore, the exact mechanism for fast inactivation of sodium channels also remain to be structurally elucidated. Last but not least, the sodium-calcium channels represent primary targets for myriads of toxins and FDA-approved drugs. So, for example, in sodium channels, 7 primary acceptor sites have been identified for responding to toxins from different venoms of animals. So because of the asymmetric nature, none of these sites can be modeled based on the structure of homotetrameric VGSE members like potassium channels or bacteria homologs. For this reason, we set out to solve the structure of eukaryotic sodium-calcium channels. On the other hand, even despite the high sequence similarity of these channels among themselves, there are quite some different properties associated with the complex formation of these channels with different subunits and also the variations in sequence. For example, the 10 mammalian calcium channels are classified to 3 families like CB123, and they certainly have distinct electrophysiological properties. So are sodium channels. The sodium channels are even more close to each other. However, they do exhibit different sensitivities to toxins or different kinetics or dynamics for inactivation, fast inactivation. In particular, sodium channels possess more than 1,000 point mutations that are associated with various disorders. So many of them are important drug targets. For example, NaV1.7 and 9 are well-established targets for development of next-generation painkillers. However, when we try to design inhibitors or antagonists targeting these channels, you may want to avoid NaV1.5, the cardiac subtype. reveal the distinct biophysical properties of these channels and to contribute to drug discovery. Instead of focusing on one particular sodium or calcium channels, we had adopted a rather aggressive approach to systematically characterize all representative members of human sodium calcium channels. So in the following, because this is a celebration of like PDB for 50 years, instead of focusing on our recent progress, I would also like to give you an overview, a brief overview of our work in the past decade. So thanks to the revolution of cryo-EM, we were able to solve the structures of a number of eukaryotic calcium sodium channels. Like we started with the largest ion channel random receptor, as a training for ourselves. Of course, they are also important and they share the VGIC superfamily fold. And then after getting more experience with cryo-EM, we were able to finally solve the structures of eukaryotic sodium channels. The first from American cockroach because of its better solution behavior, followed by the archetypal channel from electric eel. So this channel was actually the first sodium channel to be cloned and to be biochemically characterized. Well, it's a pity that it was actually the second sodium channel to be structurally revealed. And after that, we were more experienced and we were able to solve the structures of human NAV 1.1, 2, 4, 5, and 7. So as I said, our goal was to reveal their distinct properties and to summarize, some common features. To understand the molecular basis for common mechanisms. So here's what we've learned from these structures. Because of the time limit, I will just go through quickly without details. But you're welcome to present your questions, and we'll try to reply either live or through email. So I think so this picture was prepared many years ago to exemplify the power of cryo-EM. So we didn't realize that we were able to see different shapes of calcium channel, different shapes of the density in calcium channels when we apply calcium chloride in different conformation. In different concentrations as shown here. When calcium was applied at 10 millimolar, you can see actually a stretch of density lining the selected filter of CAV 1.1. So we deconvolute this density to 2 ions. And then when we reduce the concentration to 0.5 millimolar, the density becomes like a spherical shape. So this may represent this high affinity site for calcium channel. And with regard to sodium channels, so here, the all 4 amino acids DEKA are well resolved, and we were also able to identify a density that is, surrounded, that is caged actually by 3 carboxylate group. Including the 2 from DEKA motif, D and E, and also an invariant glutamate from the second repeat. So we define this site as the DEE sodium binding site. So this structural observation is also consistent with our MD simulation analysis. As you can see, based on the structure, MD simulation reveals a potential sodium permeation path through the selected filter. And now let's move down from the selected filter to the intracellular gate. So although we solved more than a dozen sodium channel structures, so far we only captured 2 primary conformations with some subtle variations. So we cannot claim any of them to be open despite the different radii in the constriction site, the intracellular gate. Nevertheless, the dramatic difference in the diameters of the intracellular gate did give us a hint on the potential conformational changes associated with the gating. Shown here is an intracellular view. As you can see, the central pore domain does undergo an iris-like rotation for the tetrahelical bundle of S6. More importantly, each S6 helix actually also undergoes an axial rotation that results from alpha to pi helical transition. So actually, this structural feature has been observed also in trip channels and calcium channels. So we think this, secondary structure transition is an important element in, the gating of VGSC members. Apart from this intracellular gate, the sidewall actually is also gated. You can see, the sealed ones are from the structure of nav pass, which is the sodium channel from American cockroach, whereas the ones with the hole, which we name fenestration, belong to the structure of the one from electric eel or like human channels. This observation is also important because it is known that this particular site is for anesthetic binding, and the conformational change is also consistent with the previous analysis that the local anesthetics only bind to the channel when they are activated or inactivated, but not at the resting state. So this structural observation also suggests that the structure of nav pass, which has sealed fenestrations, may represent a potentially pre-open or resting state of the pore domain, but not the voltage sensing domains. But I cannot say, that really reflects the gating process because intracellular gate is not completely open for ion conduction. Then, the most exciting structure discovery actually came from, the structure comparison for fast inactivation. So let me remind you, the linker, a hydrophobic cluster, IFM motif on 3,4 linker is responsible for fast inactivation. And, a traditional or like classic ball and chain model is like this, this motif may be inserted or maybe directly block the intracellular gate. So interestingly, this feature was captured only last year by cranial imaging in Weill Cornell in the study of MTHK. However, so when we saw the structures of nav pass and the calcium channel in 2016-17, this 3,4 linker was observed to be sequestered by a globular C-terminal domain. And here, if you can see my cursor, here is where the IFM motif will be positioned. And the structural analysis shows that it is impossible to place this IFM motif directly below the intracellular gate. So the bond and chain model may not be correct. And then just a few months later, after we solved the structure of sodium channel from electric eel, we did observe a dramatic conformational change of the 3,4 linker and the IFM motif. You can see, this IFM motif is repositioned to be inserted into the pore domain. But does it mean that the bond chain model is correct? Actually, if you examine the position of this IFM motif, instead of directly blocking the intracellular gate, it is inserted into a cavity outside of the S6 helical bundle. It is enclosed by the S6 helices in repeat 3 and 4 and the S4 and S5 restriction ring. So in a sense, this reminds us, this IFM motif reminds us of a door wedge. So to close the door, you can either block it or you can use a door wedge to squeeze it to close and to prevent it from reopening. This structural observation was truly exciting, and more importantly, based on this structure, many previous functional studies can now be interpreted because many residues, previously characterized residues, are either directly involved in the on the linker, or they actually map to the acceptor site on this pore domain helices or S4, S5 rings. So this model, which we proposed, the door wedge allosteric blocking mechanism that we proposed in 2017 based on the structural comparison of nav pass and the one from electric eel, was recently structurally reviewed by the beautiful work from Nieng Yan and Bill Ketter's group. So when they replace the IFM motif by a triple glutamine, you can see the density is no longer visible. and accompanying the disappearing of this density, well, this corresponds to potential flexibility. The S6 helix in repeat 4 also moves outside. So in a sense, indeed, insertion of the FM motif into this cavity pushes this helix inward to close the gate. So this is indeed door wedge to close the door. So this is really gratifying. All right. So after here's another part, like the mode of action of the drugs and animal toxins revealed by the structures. I will not go into details. So I just have one slide here to show how powerful cryo-EM is in elucidating the detailed interactions between these small molecule drugs or toxins with the channels. So I think this slide can be expanded to a 1 hour talk. I will not go into details. Sorry about it. Okay, despite this exciting progress, there's one essential question remaining. That is, the molecular basis for the electromechanical coupling of the voltage gated ion channels. So despite our extensive effort, there are, as I just mentioned, there are only 2 primary conformations captured for all the sodium and even calcium channels. So that of the one from American cockroach, NavPass, and that of the one from electric eel, which we named EE-NAV 1.4. Basically, almost all the human sodium channel structures are almost identical to the one from electric eel, which we think represents an inactivate conformation. So structural comparison of NavPass and other channels did provide some hint to the potential electromechanical coupling process because of the different conformations of VSD. But these conformational changes are relatively minor. I mean, there can be like a 1 or 2 residue transfer, and more importantly, they come from the channels with like lower sequence similarity. Ideally, we hope to capture the structure of one specific channel in different states. Well, we have been actively working on that. Here came this exciting breakthrough in computational biology, like by AlphaFold and Rosetta Fold. So we were curious whether this predicted structure may yield different conformations. Well, in terms of sodium channels, I have to say, all the predicted structures are almost identical. Well, as the name indicates, they reveal the correct fold. Maybe because they're trained. I mean, the training is based on the almost identical structures. Therefore, so far we haven't been able to benefit from the predicted structures for revealing the electromechanical coupling process of sodium calcium channels. And apart from us, like several groups also tried actively to capture different conformations, such as, show here 2 examples, like Jian Peong from Genentech engineered a chimera of NavPass and a VSD4 from NAV 1.7 by applying a toxin. VSD4 is captured in the down conformation. And Bill Cateros and Nieng Yan's group also applied a potent toxin to NAV 1.4 to lock VSD4 in the down state. However, despite these 2 different conformations, there is literally no conformational change of the pore domain. Therefore, the electromechanical coupling process is not revealed. So we, my group is also, trying different approaches such as extensive mutagenesis or application of different modulators. Unfortunately, so far, no additional conformations have been captured. Well, we are working with sodium channels, in the hope to capture different conformations. A serendipity actually came from our efforts on calcium channels. So when we saw the structure of the wild-type human CV2.2, to our pleasant surprise, the VSD2 of this channel, of this wild-type channel, in the absence of any modulators, was captured in the down conformation. So we observed a PIP2 molecule. That was completely unexpected. Probably this, PIP2 played a role in locking the structure in the down conformation. So you can see here. The conformational change is huge. It consists of both domain-wise rotation and the secondary structure transition of one particular helix S4. And you can see, the secondary structure transition for S4 on top and at the bottom, accompanying the gating charge transfer, as shown here. All right, I see I ran out of time. So this is also, this is a wild-type protein. We then, searched, the predicted database by AlphaFold2, and so this time again, the conformation actually predicted by AlphaFold2 is similar to the previous, app states. It's not consistent with our experimental structure. Therefore, we think, there's still some space for the, AlphaFold or Rosetta fold to improve to, capture different conformations of the sodium channels. So I still have several slides for our ongoing effort for capturing different working states of these channels, but I already ran out of time, so I can save that for question session. So with that, I would like to conclude my talk. Okay, let me just jump to the acknowledgements slide. This is actually a summary of the 1 decade of work by my talented team in Tsinghua prior to 2017, and then in Princeton. So here, show here are quite a few chemists and material scientists. So that shows our effort to combine different methods to log these channels in different conformations. But we'll see. This is the kind of we can have, a competition with the computational team to see who would reveal the complete cycle of these miniature molecular machines. And thank you for coming to this seminar. I'm ready to take questions.
Thank you. Thank you, Nieng. It was great. One of the particularly exciting things about membrane proteins, and your systems really make the point, is how conformationally diverse they are and how that conformational diversity underlies their functions. I'm curious whether in crystallography, you crystallize one form and then maybe you add or change the conditions or add a ligand, you crystallize another form. In cryo-EM, you can see multiple conformations in the same sample. So in your case, are you seeing that? Are you able to detect multiple conformations and use class averaging to get multiple structures? Or are all of these selected in some way?
This is a wonderful question. So in our previous efforts, we maybe because of our lack of experience, we basically identify 1 or 2 primary sequences. But then just in the past 1 year also, these results are not published. We even reprocessed the previous data and identified new conformations. So yes, you're right. Now, just within one data set, we can obtain several conformations. This is, even more exciting than before.
Yeah. So maybe this actually would lead into another question here, which is from H. Raghar Aman. Despite numerous wonderful structures solved, what could be the reason for not capturing all the conformations you need of the sodium channel to address the cycle?
Yeah, this is a great question. Actually, I did prepare a slide. So because there can, this is continuous process, right? In terms of conformational change, it's a continuous spectrum. But we can artificially divide that into 3 major states: resting state, activated state, and inactivated state. So most of the structures represent the inactivated states. Why? Because that's energetically most stable. That represents an energy minimal. For resting state, just think about it. So in the excitable systems, a strong electric field is required to hold that conformation, right? Minus 70 millivolts across just 3 to 5 nanometers. That's a huge electric field. So that's why, the resting state without this external electric field is not stable. And then for the activated state, it's also transient. It's a transient conformation. So that means it's not stable either without, interference by modulators or, this kind of, I think, sometimes I imagine the active state as a spring just transient thing. So I think, it's all about protein folding because, to capture those states, you need extra energy. So this equilibrium can be shifted either by addition of modulators or by mutations to shift the, the kind of the equilibrium, or by applying an electric field. So that's our major effort. We try to introduce the electric field, directly introduce electric field by engineering different systems.
A couple more questions. So Nina Pastor asks, do you think that the opening mechanism of domain-swapped voltage sensors will be different than those of not domain-swapped voltage sensors? To what degree does domain swapping affect?
That's a great question. Yeah, that's a great question because now we know there are different, there are two organizations, domain swapped, or like in cis or in trans. So if I understand correctly, if you refer to the conformational changes for the pore domain, I would think, the paradigm should be similar, like, the Aristotle rotation or the helical rotation. But in terms of the driving force for, the specific mechanism, there can be variations.
And okay, I'll read another question. Question about fast inactivation. When studying IFM affinity to its elusive binding site, how has the temperature of the cryo-EM changed that site?
So I'm not sure whether I can address that question because of cryo-EM. So basically, theoretically, you freeze, the state of your molecule at so this is a very fast thing to avoid formation of ice, right? The crystal ice. So basically, the temperature of cryo-EM, if you mean a cryo condition, we hope that does not alter the state so that the structure can reflect the solution state, like 4 degree or 20 or the room temperature.
So we'll do one more from Kathleen Eiter-Jeets. In your recent calcium channel structure, how do you explain the functional relevance of the outward conformational movement of VSD2?
So thank you for reading our paper. Yes, in our CV 2.2, we see like this VSD2 in the down conformation. And it is like, the entire channel is in the, especially the pore domain, seems like, it's even more tightly closed, lacking the fenestrations. So we think like this state, we agree, actually we also performed some functional analysis. So we agree that this state may represent the so-called closed inactivation state.
So Nieng, there are a few more questions we didn't get to in the chat, so you'll be able to interact with people offline by email. So thank you very much, and especially the illustration about the conformational diversity and how it underlies function is very beautiful. Thank you.